My name's Will Duguid. I'm a postdoctoral researcher at the Juanez Lab at the University of Victoria. My primary work is investigating the ecology of juvenile Chinook salmon in the Canadian Salish Sea. But today I'm going to talk a little bit about some Pacific Salmon Foundation supported work on the diets of adult salmon in the Salish Sea. So Jacqueline's already done a really nice job of explaining what forage fish are and explaining that forage fish provide the linkage between the small zooplankton at the bottom of the food chain and the large predators at the top of the food chain. An interesting aspect of marine ecosystems is that they've been often described as a wasp waste ecosystem. And this refers to the fact that often it's a relatively small number of forage fish species that form the linkage between much more diverse communities of zooplankton prey and predators. And this wasp waste refers to the shape of this food web with low diversity in the center at the waist. And because there's relatively low diversity of forage fish, it means that changes in the composition of the forage fish community at the waist of the ecosystem can have really major implications for ecosystem function. So this is why it's very important to understand what's happening with forage fish and how their abundance is changing and how species composition is changing. So how do we study marine ecosystems? How do we investigate what's actually going on in an environment that can be hard to see? Traditionally, this tends to involve large research vessels conducting fishery independent surveys. And so the vessel pictured here is Canada's new Pacific Ocean Science vessel, the Coast Guard ship Sir John Franklin. This sort of work is not cheap. The Franklin probably costs in the order of $30,000 a day to operate, and that's when it's working. So while these sort of surveys, these fishery independent surveys conducted by government agencies provide really, really valuable information, the costs of these programs and also the other demands on these research vessels mean that there's often significant holes in spatial and temporal coverage. These large ships can't be everywhere all the time. So what other options do we have to fill in some of the holes in the insights provided by traditional fisheries science? So in British Columbia, we have a year-round public fishery. And I have a long connection to the salmon fishery in BC. I grew up salmon fishing as a child on Vancouver Island. And actually, between my undergraduate and my master's degree, I spent 4 years as a fishing guide in Campbell River. And I still try to get out there as much as I can. And as a participant in the public fishery and talking to recreational fishermen over the years, I came to realize that there's a terrific amount of knowledge held by the recreational fishing community about where salmon feed on different types of prey, when they feed on different types of prey. And as I developed my career as a fishery scientist, I realized that a lot of this information, while it may be common knowledge to fishers, is actually not common knowledge to fisheries scientists. And I realized that all of these salmon stomachs that are being used as crab bait or going down the hole at the cleaning station, like the one pictured here, potentially are a source of some very, very valuable information on the ecosystem and how it functions. And that idea kind of became the origin of the Adult Salmon Diet program at UVic. So in 2017, we started to reach out to anglers and say, would you be interested in submitting the stomachs of the salmon you catch to a program at UVic? And we had some pretty good uptake. People were enthusiastic about the idea. And so we decided to move ahead with it, and we framed some objectives for the program. So one of the first short-term objectives was just to fill in some basic knowledge gaps about coho and Chinook salmon diets in British Columbia. One would think this would be pretty well understood and well known in the scientific community, a pretty low-hanging fruit, but in fact, no research had been published on adult Chinook or coho salmon diets with data more recent than the 1960s. Another critical short-term objective was to improve the understanding of a forage fish ecology. So basically, to improve the understanding of what's happening at that critical wasp waste of the Salish Sea ecosystem. And then in the long term, a goal was to use the salmon diets to monitor ecosystem change, to actually ask the salmon how they were perceiving the environment. Because in the context of a changing climate, it's quite likely that the relative abundance of forage fish at the waist of the food web will change. And this would be an excellent way of monitoring that change. And then we also wanted to develop and maintain a citizen science network. And we saw fostering that two-way communication between fishery scientists and fishers as an objective in itself. So in developing this program, we had to come up with a way of engaging anglers. And we pursued this a few different ways. We reached out through online sport fishing forums to encourage participation. We actually attended fishing derbies and met anglers, chatted with them, cleaned their fish, took their stomachs, and tried to get them to participate in the program. We also reached out through social media. We wrote a number of magazine articles, like the one pictured here at the center of this slide, trying to stir up interest in the program. And once we had anglers engaged, we needed to keep them engaged. And so in order to do this, we've had a gift handout every year where if you submit 10 stomachs to the program, you receive a program hat. We also have a draw. Islander Reels and AP Tackle Works donate gift packages, and anglers who submit stomachs, each stomach is an entry in a draw. And then finally, we provide feedback to anglers. So we've developed some computer code that produces individual data reports. So you get a report back of the stomach contents of the fish that you individually submitted, along with a larger report that summarizes the results of the program as a whole each year. And as we've developed this and as we've gained participants, we've developed a pretty good sample pipeline. So we provide these waterproof data cards on which anglers can record the catch location and date and other details of the salmon that they've caught. And then that waterproof card goes right in the bag with the stomach. And some of these collections are being managed by partners. So for example, the Pacheedaht First Nation on the west coast of Vancouver Island has their fisheries monitors at the dock that they manage collect stomachs and record this information from fishers as they come into the dock. We also have some guiding companies like Bon Jovi and Granville Island and Island Pursuit Sport Fishing in Comox, who have been huge participants in the program, submitting the stomachs from their clients' fish. And then a large number of our samples come just from individual anglers, and they can pick up the information, they can pick up the cards to record data, and then they can drop their samples off at depots. So these are mostly tackle stores around southern BC. And then when we get the samples back to the lab, we've developed a really nice, robust, transparent methodology for analyzing what the stomach contents are in these fish. If we used the very few taxonomic experts in BC who can identify literally the bone of any fish that you might encounter in British Columbia, this project would quickly become prohibitively expensive, and undergraduate students are much cheaper. However, high quality data is also very important. So we've developed really clear decision rules for identifying different prey types and also for determining when something is not identifiable. And when fish and other prey are badly digested, we use hard parts. And so that could include otoliths or ear bones, which are pictured on this slide here, to identify fish which we otherwise wouldn't be able to identify. Otoliths are also very useful for reconstructing lengths. So we have software that can measure a photograph of an otolith, and the otolith measurements can then be used to reconstruct the length of the fish that otolith came from, based on a relationship of otolith length to fish length for intact fish. So how has this all gone to date? The project began in 2017, and since 2018, the primary project support has come from the Pacific Salmon Foundation, which we're very grateful for. Up to this point, we've engaged over 200 participants, and we've processed over 3,000 stomach samples. Primarily, these have been Chinook salmon, but with a few coho salmon as well. From those 3,000 plus stomachs, we've individually weighed and identified over 9,000 prey, and we've trained up 10 personnel on prey ID and data management, and 4 of these folks have gone on to become graduate students in the lab, and that includes Katie Innes, who's recording data in this picture here. And what have we found? We've found lots of interesting things. In the interest of time, I'm just going to stick to a couple of highlights of the results of the program so far. So one of our questions was how do the diets of Chinook salmon, in this case, vary in space within the Canadian Salish Sea? And to get at this, rather than just looking at how diets differed between arbitrarily defined regions, we actually went to the data and said, what does the data say about where diets are different? And so the student who led this analysis was Wesley Greentree, who's a wildly capable undergraduate student who actually did this work for his honors thesis. And what Wesley found is that there were 4 regions within the Canadian Salish Sea that were characterized by different diets. The Strait of Georgia and the Strait of Juan de Fuca were characterized by being overwhelmingly dominated by herring. And while herring were important in all regions, in the area around the Lower Mainland and Howe Sound, anchovy were also extremely important, but not elsewhere. Whereas in Harrow Strait and the approaches to Juan de Fuca around Victoria, sandlance were extremely important, but again, were less important elsewhere. So the importance of northern anchovy around the Lower Mainland is really interesting. Some of you may remember that around Christmas 2 years ago, there was a bunch of news stories about this huge abundance of anchovy that appeared at the White Rock Pier, and people were coming from all over the Lower Mainland to watch seals and sea lions and birds feeding on all of these anchovy. So this is really interesting. This is just one of a number of observations of really abundant anchovy around the Lower Mainland in the last 5 or 6 years. And we've done some research that suggests that the abundance of anchovy in this area has actually increased with warming waters and potentially with climate change. And so we're really, really interested to see what our program tells us going forward about changes in the abundance of anchovy within the Salish Sea. The importance of Pacific sandlance in Harrow Strait and the area around Victoria is not surprising, because we know that this is an area of excellent sandlance burrowing and spawning habitat. What's interesting is the lack of sandlance and diets in other areas of the Canadian Salish Sea that also have some quite good spawning and burrowing habitat. So we're working with Jacqueline, World Wildlife Fund Canada, and Cliff Robinson and others at DFO who are interested in these issues of sandlance habitat. And we're really hoping that going forward, our program will be complementary to the sort of efforts that Jacqueline has described to monitor and preserve forage fish spawning habitats and understand the role that those habitats play in the function of the ecosystem as a whole. Now, I shied away from too many graphs in this presentation, but I couldn't resist showing this one because I think it tells a really compelling story. So this is an index of the fullness of Chinook salmon stomachs in the 4 regions in the map at the top right. And it runs from 2017 on the left to 2021 on the right. And the colors of the points line up with the colors of the regions in the map at the right. And so with the exception of 2017, when we were just beginning the program, when we didn't have that many samples, a really clear story emerges. And there's 2 points. One is that the Strait of Juan de Fuca appears to be an area of consistently low stomach fullness. And another is that the Strait of Georgia appears to be an area of consistently high stomach fullness. So it seems that at least in these years of the program, the Strait of Georgia is a really good place to be if you're a Chinook salmon. So why might that be? And it turns out, I mentioned earlier that we're able to reconstruct the length of the prey in these salmon stomachs. And it turns out that about 50% of the summer diet of Chinook salmon in the Strait of Georgia is large adult herring. And the reason this is interesting is because the Strait of Georgia herring population is generally considered to be a migratory population. That is, while they spawn in the Strait of Georgia, the vast majority of the population migrates out of the strait to the continental shelf on the west coast of Vancouver Island to feed in the summer. And what our work has shown is that it is actually within the Strait of Georgia in summer, there's quite a lot of adult herring that haven't migrated, and that these adult herring are extremely important for the ecosystem. And so Jessica Qualley in the picture here is a master's student in our lab who also started working on the Adult Salmon Diet program in our lab. And she's now doing a project trying to understand what the factors are that control the abundance of these resident herring in the Strait of Georgia. And I think this is a really key question for the health of the Strait of Georgia. And I'm really excited to see how Jessica's work turns out. Some of that work is also involving samples derived from the Adult Salmon Diet program as well. And I mentioned that one of our goals is also to monitor change, to assess how that key wasp waste of the ecosystem may be changing through time and changing in response to climate change. And so again, this is another graph, and we've only had this program going since 2017, so it's early days for monitoring change. But I wanted to show you this just to give you an idea of the sort of thing we hope to be able to do. So here we have a bar chart that the total height of the bars represents the stomach fullness of Chinook salmon, and the color of the bars represents the contribution of different prey types to that fullness. And this is an index that's balanced to compensate for different numbers of samples in different seasons in different regions in each year of the program. So what we see is that stomach fullness is pretty constant across the years of the program so far, suggesting that fish are finding a similar amount of prey. Perhaps the importance of herring is actually increasing here, moving from 2017 to 2020, and the importance of anchovy and light orange is decreasing. But this just gives a sense that as we move forward and as the ocean changes in response to changing climate, if species drop out of the system, if new species appear in the system, if salmon suddenly can't find enough food, this program is going to provide really powerful insights to how those changes are occurring. So I think I'll leave it there for today, but I want to thank the funders who have supported this program, and particularly Pacific Salmon Foundation, who provided the primary funding for the program. And I'd also like to thank all of the folks who have done a lot of the dirty work. So a number of folks who've done the smelly and messy laboratory work of processing all these samples, a number of the collaborators who we've worked with, the depot locations where anglers can drop samples, and then also really importantly, all of the participating anglers who've taken the time to do this extra paperwork at the end of their day of fishing and submit these samples to us. And I'll hand it back to Jacqueline, who's going to talk a bit more about the conservation and protection of these important, ecologically important forage fish habitats.